Alright, pre-AP, we're going to hit the highlights on protein synthesis. This is going to be transcription and then translation. Please make sure you know the difference between those two words. Transcription, translation. Make sure you got a highlighter and a pen. We're going to highlight the things that we really need to know and break down this big old chunky outline into parts that we can use. So, let's get started. The first part of this is going to talk about a guy named Beetle, as I call him. His name might be Biddle. I still like to call him Beetle. And then Tatum. They first said that one gene coded for one enzyme. Nope, nope, nope. We don't need to know that. We need to highlight the very last sentence where it says one gene, one polypeptide. Remember, polypeptides are proteins. So one gene, one polypeptide hypothesis, because enzymes are a type of polypeptide. So just know that every gene, every segment of DNA codes for one polypeptide, one protein. And just a side note again, enzymes are a type of protein. Alright, so transcription and translation. I'm just showing you the difference between prokaryotes and eukaryotes. The top one is prokaryotes. Their transcription and translation is really, really simple. If you're a eukaryote, it's a little bit more complicated. We're not going to talk about these guys, though. We're going to mainly focus on them. We're going to talk about us, eukaryotes. Alright, so let's look at transcription. Let's highlight some things real quick. Transcription, please highlight. It's the process of making a working copy of an original. Out beside original, put an arrow and put DNA. Out beside transcription, real quick. Let's make a note. Remember we talked about making pancakes. Out beside transcription, put you a little bitty star and put what we're going to do to that recipe next. In replication, we copied the recipe. Now what we're going to do is read the recipe. We're going to read the recipe. We're going to take DNA, so write this down too. We're going to take DNA, we're going to read that DNA, and we're going to make something called mRNA. That little M is for messenger, if you want to put that out there so you'll remember. Oop, I so misspelled messenger. Let's see, redo that. Messenger RNA. So let's highlight the process of making a recyclable working copy of DNA in the form of RNA. We're going to talk about a specific RNA that it's going to make mRNA, messenger RNA. Messenger RNA sends the message from the DNA so we can make a protein from it. Ooh, and then in parentheses, let's highlight it. That recyclable copy will become known as mRNA. Whoa, there it is, messenger RNA. Alright, so out beside this, we're gonna put out to the side of this, put DNA versus RNA. We need to know some differences. We do. So if you want to put this is kind of like called a T-chart. Make a T. DNA. We need to know it is double-stranded. That means it has two strands. On the other hand, little bitty RNA over here, he's not double-stranded, he's single-stranded. It's like having one half of a ladder. They have a different sugar. DNA, you have deoxyribose. The sugar over here, it's just plain old ribose. And then the last thing, they have different nucleotide bases. DNA, we already talked about A, T, G, and C. Whoops, not G and G. What the doodle? A, T, G, C. On RNA, we're going to have A, U, G, C. You have to have U in RNA because it's uracil. We cannot make proteins from thymine. We can't. We can only make them from uracil. So if you want to underline the differences between those two, DNA, double-stranded. RNA, single-stranded. Sugar, deoxyribose for DNA, RNA, ribose. ATGC for DNA, nucleotide bases. AUGC for RNA. U is for uracil. Make sure you know that. Alright, so let's get back to your outline. Let us highlight that mRNA is synthesized, that's a fancy word for made, I put that out beside there, by an enzyme called RNA polymerase. This is how we make mRNA. We have another enzyme, we have a helper guy named RNA polymerase. So what we're going to do with that, we're going to make a message, that mRNA, highlight, it will be sent to the construction site called a ribosome. Because remember, if we remember back to cell unit, what do ribosomes do? Whoa, they make protein. For building proteins. Ooh, highlight again, RNA, nucleotides. Use ribose instead of deoxyribose as their 5-carbon sugar. And highlight number 4, RNA. Again, you get uracil. It replaces thymine. Thymine can't exit the nucleus, those nuclear pores. So, it has to have uracil. And then number 5, mRNA is a single-stranded molecule. Because it's single-stranded, it's less stable. So we have to go do something with it. Please highlight big letter B. DNA serves as a template for making mRNA. So let's make us one, shall we? Let's say that we have DNA, just makeshift DNA. We'll say there's A. We'll spread these apart. A, T, G, G, C, C, T. That's our sequence. That's the gene we want. We want to make some mRNA from it. Let's change our color so we don't get confused. So, mRNA. Here, this is what we do. Instead of A pairing with T now, it's going to pair with U, uracil. Now, if we get to the next nucleotide, you're thinking, whoa, Miss Minor, there's a T there. Does T go with U? Does T still go with A? What do I do? It still goes with A, don't worry. Go ahead, put this as your little cheat sheet up here. A equals U. T equals A. T cannot, that's why we put a slash through it, cannot equal U. Ta-da! Again, what does G go with? Good job, C. C goes with G. And then again, just to give you another practice, what does T go with? That's right, it goes with A. Good job, guys. This is basically what transcription is. We take our template DNA. We take that pancake recipe. We see all the milk, the flour, the sugar, the eggs, the vanilla, and we're going to actually read that DNA, transcribe the DNA, and make us some mRNA. We're making a message so that we can go make protein in a minute. So please keep that in mind. We're going to do an activity with this. Alright, so transcription, translation. To transcribe means to read, to make sense of. We want to make sense of that DNA so that we can get a message out to the ribosome so he can make protein. And that message is in the form of mRNA. So if we look at our picture down here, transcription, translation. We already put for transcription, we're going from DNA as a template and making mRNA. Out beside where it says translation, that big old Roman numeral number 3, let's write this out beside there just to make some more sense, guys. Make mRNA. We got it from transcription. Whatever you get from one step, you are going to use in the next one. Let's move this down here so we don't get confused. mRNA, then we are going to make protein. That's what we're going to do. We're going to take that message and we're going to make our final product, protein. If we look at our picture down here, we have DNA. We took and transcribed it. We read this DNA. Remember that guy who comes and reads the DNA? Remember his name is RNA, poly for short. Remember he's an enzyme. He comes and he reads this DNA and he matches up RNA nucleotide bases. He puts A with T, C with G, all the way down and makes this lovely mRNA strand for us. We can use this now to go out into the cytoplasm where the ribosomes are and make protein. Translation. Translation means to make something. We are translating and making sense of something. We're going to take this mRNA and we're going to make more sense of it and form an amino acid chain below protein. Please write this. Amino acid chain. Because remember, the monomers for proteins are amino acids. We're going to make a chain of amino acids which will fold up and make a protein. So the next thing we need to know just remember Shergoff's rule. We're going to have a math quiz on this in the morning. Make sure you go back and look at DNA history. Marshall Nuremberg, I'm not going to ask you really about him, so don't worry. RNA codon chart. This is where we got to. This is Roman numeral 4. Please highlight codons, aka triplet code. We need to know that this is the RNA language that will be translated into polypeptides. Remember, polypeptides are just a string of an amino acid chain, which make proteins. We need to know that codons are 3 letters, or 3 nucleotides of RNA. Remember, we got that RNA from a strand of DNA, so let's make an example. Let's say, remember, codons are 3 letters. I like to think of this as this is your zip code. Pardon me, not your zip code. This is your area code. What is our area code, ladies and gentlemen? It is 256. Any phone number you want to call, 256. Codons can be anything. They could be U, A. That's just a random one. We're going to take this codon, this 3-letter sequence, kind of like your area code, and we're going to plug it into this codon chart to find out what amino acid it makes. So make sure that you pay attention how I do this. First base, first letter, you're going to look over here. Oh, it's a U. Oh, there we go. Found my U. Now I can move on to my second base. Well, let me find it up here. Oh, a U. And then I can go over here to my third base. Oh, well, where's it at? I don't know. Oh, there he is. 2, you can kind of cheat. You can just look through here and go up U-U-A. Some of you probably already figured that out. You're kind of thinking, Miss Minor, I already found it. That's okay. First base, second base, third base, home run. So codons, 3-letter sequences, 3 nucleotides, code for an amino acid. The name of that amino acid is actually leucine. Alright, so let's continue on. What we need is go to big letter B. We don't need anything else from A. It's okay. Move on to B. RNA codon chart for amino acids contains 20 known amino acids. There are 20 known in living things. We need to know 3 things. Just know that RNA is referred to mRNA. Stop there. Big letter B. Each 3 letters, or 3-letter codon, codes for 1 and only 1 amino acid. That's the big one. Please highlight that most amino acids have more than 1 codon. That's okay. And the big one, put a star by letter D, the genetic code is redundant. You can have U 3 times. And then you can have 3. If you had 3 U's, it's okay. That just means you have 3 phenylalanines. It's okay, it's redundant. Please highlight that number 3 down there. AUG is the start codon. And that it codes. It is the codon for the amino acid methionine. Anytime you start an amino acid chain for a protein, it only starts with methionine. And then number 4, UAA, UAG, UGA are stop codons. Number 5, this chart is universal for all living things and viruses, even though viruses are not considered to be living because they must live inside a host to survive. If they exit a host, then they die. And then number 6, the codon will match an anticodon. So let's make an example of that. An example would be your codon. Let's just use U. Your anticodon. What would be the opposite of U? What goes with U? Please say A. Your anticodon would be that. Anti means opposite. And then reading frame. Just know that this returns to a set of 3 consecutive nucleotides. It's just reading frame means that we're going to read this mRNA strand 3 letters at a time, 1 nucleotide at a time. Come on, little fella. Work for me. Alright, so we're on to part 2. Then there will be a separate video for part 3 and part 4. This is going pretty quickly, guys. So, mRNA synthesis and modification. Please just highlight this is the making of mRNA. This is the process of transcription. So put you a note out beside there. Again, this is taking DNA and turning it into mRNA. We are going to read the recipe, so just write that out again. Taking DNA, making it into mRNA. Making some shortcuts. So again, turning DNA code into mRNA code. So highlight there are 3 phases of production. Initiation. Please highlight it's the building or factory to make mRNA. We need proteins to make proteins. It's crazy. So little letter A, a protein called transcription factor attaches to the promoter sequence. Where you see the word promoter, put yourself a little arrow and put start here. That's all that means. Promoter means it's almost kind of like a cousin of primase. He stands there and goes, start here, start here, start here. He has a little flag. Make it checkered. Start here, guys. A promoter sequence of the gene to be transcribed. Please know in little letter B, this whole factory, this transcription factor that attaches to the promoter. So here he is, transcription factor. He starts at this teeny tiny little segment called a promoter. And this thing together, those two things together, make a transcription initiation complex. So just highlight that, please. I know you look at these words and you think, that's crazy, Miss Minor. It's okay. You just put in, see, here it is, transcription initiation complex. You have your promoter. You have your transcription factors. RNA polymerase is going to come in here in a minute. Elongation. Please highlight and put out beside there, please. This is all elongation means. These words look really intimidating and scary, but elongation just means you're going to stretch out and stretch open your DNA, and you're going to add nucleotides. You're going to add A, U, G, and C to form this mRNA from a DNA segment. So please know this refers to actually making RNA. And highlight, all you got to know is little letter B. RNA polymerase separates the DNA to make room to work and adds nucleotides to the growing molecule. That's it. That's all you got to know. Termination. Remember, what does terminate mean? To stop. So, just like it says, we're going to stop transcription, so highlight that. A stop codon is made. Ooh. And the factory molecule slows down. Please know, little letter B, RNA polymerase slows down until it stops transcription by forming a, this is true, AAU, AAA sequence. And then it's released from the DNA. You don't have to try to memorize that sequence. It will not be on the test. It's just to know that, hey, whenever it gets to this point, a specific point, it will stop. Oop. Post-transcription modification. Remember, to modify something means that we are going to make it better. So what happens is, for nut number 1, please highlight that little letter A. 5' protective cap is added. It's like you putting on a hard hat. We're going to take this. So go ahead, just draw this little structure right here. Out beside there, you have room. There's going to be, this is mRNA. You're going to put on a 5' cap. It's going to protect your segment of mRNA. Because if you just exit it out into the cytoplasm, well, it could disintegrate. Number 2, on the back end, right back here, we're going to add something called a poly A tail. So please highlight that. That's all you got to know. It's there to protect it from digestive enzymes in the cytoplasm from destroying this lovely message. And then number 3, what happens in the middle here? What we need to do, we need to highlight. During this step, we remove all non-coding introns. These are little segments that we don't want and we don't need. We don't need them. And we do this by using something called a spliceosome. To splice means to cut up. And we need to highlight that the spliceosome is a type of enzyme. He does. He acts like scissors. He comes in and he snips out all these little introns and gets rid of them because we don't need them. If you don't need them, get rid of them. And then he rearranges all of these lovely exons. These are the part that we need. And then, ta-da! You cut them out, you put them together, and then you form that guy. So, the spliceosome is also going to stitch whatever exons are left and make us a nice, pretty, modified piece of mRNA. And then you're ready to go. Alright, that's done with part 1 and 2, people. Let's head on to part 3 and 4.